I would like to turn over the mic to Franz Naberhaus to introduce our first VAAM award today.
Okay, so hello from my side. I think that was a fantastic opening session, so it was a good start for this meeting. Thanks to Antje and Christian. That was great and really fascinating. And we're moving from highlight to highlight now, and the next highlight is the VAAM Forschungspreis or Research Award. And as you all know, that's an open call, and we received 5 nominations for this prize this year, 4 from academia and 1 from industry. And we were impressed by the quality of all the nominees. And we decided then to establish a new prize, the Innovation Prize, that will be given to Birgit Hoff later on. So that's going to be the next highlight here of this session. But for now, it's my pleasure to introduce and congratulate our Research Prize winner, Anastasios or Nassos Typas. There he is. Hi, Nassos. Hi. And so he is in Heidelberg, and he will give his talk from there. And I would like to introduce him first. Nassos studied in Thessaloniki biochemistry in Greece, and then he came to Germany to Regine Henge's lab in Berlin, where, no surprise, he studied Sigma S with Regine, and they together published 7 papers. And after that, he moved to San Francisco to join Carol Gross's group, and there he established a number of really powerful and very clever high-throughput techniques that he then took along to Heidelberg, where he is in about 2011, and where he started as a junior group leader, and now, as I learned from our recent communication, got a permanent position as senior lecturer just a few months ago on synthetic microbiology. Nassos received various prizes and many highly prestigious prizes, and I just want to mention the ERC Consolidator grant that he received in 2018. And there were 2 reasons why we were so impressed by his work and why we decided to award him this research prize. The first reason is, of course, if you check PubMed or other resources, is the high quality of his research. So he published more than 50 papers, and many of them in very shiny journals like Nature, Science, Cell, and so on and so forth. And that also reflects the quality of his work. So we were impressed by, in particular, that he does not just use high-throughput methods, but he also uses them to extract fundamental information on fundamental biological questions, such as cell wall biosynthesis, host microbe interaction, the microbiome, and also recently now microbial resistance, antimicrobial resistance. So that was one of the reasons. The second reason, I think is also equally important, is that Nassos is very enthusiastic about mentoring. So although for almost throughout his entire career, he did not have a permanent position himself, he has already mentored several junior group leaders. 2 of them are Anna Buchado and Lisa Meyer, who work now in their independent groups as Emmy Noether Fellows in Würzburg and Tübingen, respectively. And I think that is also something that is very, very important, even as a young scientist, to mentor other scientists and attract them to research questions and academia. So that were the main 2 reasons why we decided to award Nassos this prize. I have a certificate here that I can just show you, and that we will send to you after your lecture, that will come with award of 10,000 euros that will be sent along, but only after you have given your presentation, of course. You need to earn it. And so I'm very much looking forward to your presentation, and I'm sure you will all convince us that it was the right choice. Nassos, go ahead.
All right. Thank you, Franz. Thank you, Christine. Thanks to the committee for the award. Hi from Heidelberg. I'm sorry I cannot be there, but it's the current situation doesn't allow too many things. All right, so I'll start the talk and I'll give you a little bit of an idea of what we do in the lab, but I thought I will start with the most important slides in the beginning, so in case I run out of time, I don't have to rush them at the end. And this is basically the reason that I have this award. So I first have to say that awards are usually given to individuals, but at the end, science is a teamwork. And I have been blessed in the last 9 years at EMBL to have a fantastic team of individuals who have also blossomed as part of the team. And the reason I'm here is because of them. So I have to thank every single one of them. And going on with the thank yous, I mean, it's hard not to mention my two mentors. So both Regina and Carol have been instrumental in what I'm doing now for science, but not only at a research level, but also at a mentoring level that Franz mentioned before. So I've been supported by them continuously in my career. So this is what I'm trying to do also for my people. Of course, without funding, we will not be able to do any of the research I will show you here. And as mentioned, science is a teamwork. We have, I don't think we publish any more papers without collaborators. If you look at our 50 publications, I think almost all of them have basically collaborators within them. So science is and new ideas come through discussion, interactions, and collaboration. So I would like to thank each single one of them. So to show you a little bit what we're doing in the lab, so the lab combines systematic approaches with molecular mechanisms. So this is a kind of agenda, this kind of a bread and butter thing that we do, and we operate at 5 different areas, which I'm showing you here. So for the purpose of this talk, I'll show you 2 things. One, I'll show you a little bit of what those approaches are and how do they get us to mechanism. And on the second part of the talk, I'll show you a little bit more on the gut microbiome, what we've been doing. So what are those approaches? So they're all based on high-throughput reverse genetics, and that's basically on the concept that you can have controlled genetic variation. You can have that as mutant libraries, where you have every individual alone or pooled, and then you can combine it with a second perturbation, and that can be a chemical or it can be a mutation. And then what you have to do is you have to have a systematic way where you measure those interactions. And the way you measure those interactions can be with many different ways. You can be measuring fitness, or you can be measuring single cell features of the different mutants. At the end, what this allows you is to basically draw connections between the genes that they're knocked out and build networks and allow you to see gene interactions, but also the whole cellular network organization. To go a little bit more specific on what I mean by that, this is actually the first iteration of what we built more than 10 years now, so when I was still a postdoc at UCSF. So what we did is that we took the E. coli knockout library. This has every non-essential gene in the genome knocked out, so 4,000 mutants, and we profiled that over 300 different chemical or environmental conditions. We can do that by measuring colony size as a proxy of fitness. So we can array up to 1,500 mutants on the same plate. So that allows us to profile so many conditions. So what you have at the end, you have a quantitative metric that allows you basically, in very simple ways, you have 4,000 mutants from one side and you have 300 different conditions from the other side. And mutants that behave the same across different conditions are usually mutants of genes that they're functionally related. And the same goes with conditions. And this guilt by association power is what gives you the biological insights. And through that, we'll be able, actually, coli has still one of the most best studied organisms, still has 1/3 of this genome of orphan genes. This is genes of unknown functions. So we're able to link 300 of them, so a quarter of them to an annotated pathway or another annotated gene. And to show you a couple of highlights there, so Franz already alluded to that. So we actually found some of the first, together with Waldemar Vollmer, so we found some of the first regulators for cell wall biosynthesis. We also saw how the machinery operates together with other machineries in the cell envelope. And we also later found also how hydrolases are regulated within those multi-component machineries. And on the other side, we actually, together with Jean-François Collet's lab, we found that lipoproteins can be surface exposed in gram-negative cells. And this happens through machinery that till then was known only to assemble outer membrane porins in the outer membrane of gram-negative bacteria, and it seems that it also pushes inside the barrel of those lipid proteins so they can be surface exposed. But what I want to show you in a little bit more detail is something more recent, and that's from a subsequent study we did, and we did similar chemical genetics profiling to different organisms. That was Salmonella, and that allowed us actually to figure out the enigmatic function of bacterial retron elements. So what are retrons? So retrons are actually the first elements in bacteria that were found, that a reverse transcriptase was found. So they were found 40 years ago, and it was a serendipitous finding in Myxococcus xanthus that it was carrying this high copy number of DNA. So of basically says single-stranded DNA, and it was a multi-copy. This is what the name is, actually, multi-copy single-stranded DNA. So in the last 40 years, what has happened is that people have actually very well depicted how those are synthesized. So there's a reverse transcriptase, and there's a substrate also that creates first an RNA, and this RNA works as a template to create a DNA. And at the end, what you have is that you have actually a DNA-RNA hybrid that a lot of times stays tethered to the reverse transcriptase. So that was what was known, but actually, and those are abundant, they're prevalent in across different bacterial genomes, but the function has remained enigmatic. So actually, based on the screen for some phenotypes that we found on those retrons, a very talented student in the lab, Jacob Bobonis, was able to elucidate what they do, and I will just show you that basically what we figured out is that the function of them, so we figured out the function based on an accessory gene that they have, all the retrons have. So this accessory gene changes across retrons, but what we figure out that this is actually a toxin. And retrons, what they are, they're actually new tripartite toxin-antitoxin systems, whereas the toxin is the accessory gene and the antitoxin is together the complex of the reverse transcriptase together with the msDNA. So this is very interesting. These are the first tripartite toxin-antitoxin systems, but at the end of the day, toxin-antitoxin systems are very prevalent in bacterial genomes. Some of the bacterial genomes carry more than 100, and still we don't know what is their physiological role. So Jacob went a step further, actually, and devised a very clever genetic screen, which allows you to identify triggers and blockers of those toxin-antitoxin systems. So what genes would activate them or would block them from acting. And he did that, and actually what he ran up is that he found many different genes that would do that, and all of those genes was of phage origin. So there were prophage, or most of those genes were actually prophage elements in the genome. So I'm showing you here one here, this is the DAM methylase, and what we know is that methylating specific consensus in this DNA creates basically the methylated DNA activates the toxin, so it inactivates the antitoxin and this way activates the toxin. So Jacob found many of those triggers and blockers, and this is what our hypotheses are basically, that those genes are carried by the phage, they activate the system, the toxin gets activated, and this leads to abortive infection. The bacterial cells don't grow, so the phage cannot propagate. At the same time, the phages have other genes, which are called blockers, which can inactivate the toxin and allow the bacteria to still grow so that they can propagate. So independently, the lab of Rott and Shorrock also find out that a completely different way that actually those retrons are indeed phage immune systems. And I think this opens a completely new field, a completely new research area. And beyond identifying how those retrons work, which they come in very different flavors across different genomes, and what genes trigger them or what phage genes block them, or how they can be used also for genetic engineering, I think there's also one more important message here, is that now we have a method that allows us to identify what are the physiological triggers of toxin and antitoxin systems, which has to remain elusive in the field. And by being able to activate those internal bacteriocidal systems, there are also ways that one can think how to exploit that to basically use them as targeted therapies. So going back to the methods, I show you the very first iteration of how those methods worked. I show you also that I alluded that we use them also in different bacterial species. What you can do is also change the genetic variation and include, instead of controlled genetic variation, include natural variation. You can have much more complex readouts, how you read the interactions, and you can also apply them not in single organisms, but on organismal interfaces. Those can be microbial interactions or post-microbial interactions. And here I just want to show you very quickly an example of the complex readouts one can use. And this is based on reading on protein activity using this method that is called thermal proteome profiling that can profile the melting curve of every single protein in live cells using proteomics. What is a melting curve? Melting curve is the properties of proteins that as you ramp up the temperature, at some point they lose their structure and they fall out of solution. So if you measure the proteins that they're still in solution, you know which proteins are still folded. This property changes depending on protein interactions and protein activity and protein state. And this is a method that actually our collaborator and neighbor at EMBL, Mrs. Savitsky, has developed for human cells a few years back. And a common postdoc, Andrea Matus, adapted it in bacteria, show that it works perfectly. You can also use it to identify mode of action of drugs. Now, what he did afterwards is that he took a part of this E. coli knockout collection. So he took 120 mutants, he grew them up, and he figured out, so he measured all the protein expression and protein thermostability in each of those mutants. So now we have 2,000 proteins. Well, we know how they change expression or they change thermostability across 120 different backgrounds. And this is an equally powerful way to associate genes together depending on their function. And this is what is showing you here. So this is a ROC curve. It's used basically to show how new data recapitulate already knowledge that we have from previous experiments. And here we benchmarking how well we recapitulate knowledge that is known for functional associations of operons, protein complexes, or metabolic pathways. And we're doing very well in each one of them. We're actually doing as well or even better than previous methods we have. But this is not only the only information you get, so you actually get information that provides info into the activity and the interactions of essential genes, something that we usually blind when you're using knockout libraries, and also provide causal and mechanistic insights in what drives those phenotypes, what changes in the protein activity and levels that would elicit the phenotypic change. Now, switching gears for the second part of the talk, I want to show you on the gut microbiome a bit on what we're doing. And to motivate this, I think the microbiome is a field that has been booming, the human microbiome, in the last 10 or 15 years. It doesn't need too much introduction. I think there we've been going on and cataloging what is the diversity of different species around, how much individuals differ between them, and here are some of the numbers. So by now that I think everybody has heard that we carry almost as many microbial cells in our body as we carry human cells, and we carry 100 times more genes, microbial genes, than we carry human genes. So all this is very interesting, and we can find who is there. And we can also build associations to health status and disease. But at the end of the time, with data-driven approaches, you can only reach up to associations. So if you want to understand further and see what is the causal mechanism or what is the underlying mechanism behind it, you need experimental setups. And this is actually what drove me to the microbiome field when I joined EMBL. So Per Bock was already here, so driving a lot of the data-driven research. So what we decided back then was that we do need the experimental platforms to test some of those hypotheses and generate new hypotheses. So what we did is that we actually collected many different species. So here I'm showing you, it's a convoluted plot, but just showing you that we have all the different prevalent and abundant species that are present on the human gut of healthy individuals, and we also have representation across the different species of the different abundant genera. And we can use that, we know how we can grow them in communities, and we can do that with automated pipelines, so we can actually measure properties both of single species, but also from complex communities. So what we did with that resource, we actually, one of the first things that we tried to look is what is the interface of drugs and the gut microbiome. I'll show you two quick stories. One is on the effects of non-antibiotics on gut microbes, and the other one is on the collateral damage of antibiotics. So the star of both actually stories is Lisa Meyer, who's started since a couple of years her own lab in Tübingen. Mihaela has actually pioneered all the microbiome work we do at the lab. So she's the first that started doing microbiome work in the lab. And Michael Kuhn is a fantastic computational biologist from Per Bock's lab. So the three of them together moved the first story. And I'll show you what the first story is and why we started. There's a lot of information out there in literature that the changes between individuals. So I forgot to say that the microbiome between two different individuals varies quite a lot. And if you try to see what factors might be behind this variation, a lot of them have to do with environment. And when you look about what environmental factors, medication is usually the number one. And there are a few examples from specific drugs, like the type 2 diabetes drug, metformin, that those effects are shown to be direct, and not only shown to be direct, they're also shown to be important for the effect of the drugs on humans, so the anti-hyperglycemic effect of metformin on humans. But this is known for very few drugs. So back then, what we decided to see is that actually whether those, what is the extent of those effects, if they're direct effects of drugs to microbes, and at the end, what is the relevance? It has to do anything with side effects, or is also part of the mode of action. So this is what we did. So we tested about 1,200 different drugs. Those are FDA-approved drugs on 40 representative microbial gut species, and we measure directly their impact on growth. And I'll just jump in directly to the results, and this is published work. So we actually saw that more than a quarter of the non-antibiotic drugs, that's more than 250 drugs, actually directly inhibited the growth of at least one microbe. And several of them actually inhibit the growth of many different microbes. The interesting part is that the effects that we saw in vitro, we could recapitulate also, we could see also in cohort data from patients, and they were also reflected in side effects that those drugs have in patients. And one last but not least thing that I want to show you also a little bit more data is that we saw that there might be common resistant mechanisms for non-antibiotics and antibiotics. And this is the plot, what shows you here. So every single dot in this plot is one of the 40 species we profiled. On the y-axis is the number of non-antibiotic drugs that had activity on those, and on the x-axis, the antibiotic drugs. And what you can appreciate is that's a fairly good correlation. So if you tend to be resistant to antibiotics, you're also resistant to non-antibiotics. If you're sensitive to antibiotics, you tend to be also sensitive to non-antibiotics. Now, this implies that if there's a correlation between those there must be common resistant mechanisms that drive this correlation. In order to test that, we actually took E. coli. We knocked out one of the efflux pumps that is known to be used for effluxing antibiotics out of the cell. If E. coli would only be more resistant to antibiotics, then you would expect basically this dot to move parallel to the x-axis. What happens is it moves across the diagonal, so it becomes equally more sensitive also to non-antibiotics. We did the same with a different species, so this Bacteroides uniformis, who had an antibiotic resistance, but Bacteroides uniformis strain happens the same thing. So it's equally more resistance also to human target drugs. We went further, so we have those libraries where they can assess the role of each gene to resistance. So what we found actually for several of those human target drugs are the same pumps, transporters, and detoxification mechanisms that have been implicated before for antibiotic resistance. So without saying that every resistant element will give you resistance both to antibiotics and non-antibiotics, I think this implies that there's a lot of commonality. And if there's commonality, a lot of those human target drugs are used for much longer periods of our life. So if they have strong selective pressure for resistance, what you might be getting as collateral damage is also resistant antibiotic resistance. So this poses the question of whether polypharmacy is a strong driver of microbial resistance. And this is what we're actually trying to investigate now as part of a new ERC grant in the lab. Now, for the second part of the small story that I want to show you, so that on the other side, the damage that antibiotics can have on gut microbes, and this is work by Lisa Meyer again, and Camille Cremans, another postdoc in the lab. And what we had already from the first screen is that we have tested 150 different antibiotics over 40 different species. This provides also quite a fine grain of how antibiotics affect specific species in the gut. This is actually missing nowadays from databases. Usually they're all summed up as gram-positive anaerobes or gram-negative anaerobes. What we saw, and I'm not going to show you data on that, is that actually the bacteriostatic and bactericidal divide completely breaks down when you test gut microbes. And this has specific ramifications. Actually, antibiotics with broad spectrum and selective microbial killing are the ones that they're most perturbing to communities. But what I want to show you here is that actually one can come up with antidotes for some of this collateral damage. And the whole concept here is that when you have a broad spectrum antibiotic, whether you can come with a second compound, and in this case, a human target drug, well, this would allow you to specifically alleviate the effect on commensal species, but allow the antibiotic to work against pathogens. And the reason we thought that this could work is because I had previous data that we were showing that actually when you combine antibiotics, the outcome is very species specific. So what we did is that we took this Prestwick library, which has a lot of FDA-approved drugs, and we took macrolides and tetracyclines. So two of the antibiotics that we had showed that had the hugest effect and that were the most perturbing communities, and we tried to find antidote effects on them. And we did find 10 strong antagonisms for each class and species. We validate all of them. Here I'm showing you the data. We showed that several of them work actually on a number of other commensal species. And we also showed that they're not affecting the antibiotic affecting pathogens. And here's just a plot to show you that. So on the y-axis, you see how well the microbes grow in the concentration of erythromycin that should be inhibiting every one of them. And on the x-axis, you see that as a function of how much dicumarol we're adding. So this is one of the antidotes. All the purple traces are pathogens that erythromycin is used for. So dicumarol doesn't change how active is erythromycin against them, but it allows a lot of the prevalent abundant gut microbes to start growing. So this works when you have individual species. It actually works also when you assemble them in the community. And it's not only working when you assemble them in the community, it actually works also on relevant personalized communities. So those are communities from 9 different individuals. And we're giving erythromycin alone or erythromycin together with dicumarol. And we're showing how many of those species are actually protected. And we saw that for the majority of the Bacteroidales, we see protection. And we can also see the same protection in mice experiments. So with that, I will stop here to stay in time. Thanks again for the award. I hope I could give you a glimpse of what we're doing. And yeah, that's it. And I'll finish also with an advertising of a conference on microbiology that happens at EMBL every couple of years and always great event. Thank you again.
So thank you, Nassos. Now you earned the award and also the award money that will be sent to you. And thank you very much for sharing some of your data with us. I think it was pretty evident that it's really exciting research and that you deserve the prize. And good luck for all the new projects that you just had us look into. Thank you very much. All the best.
Thank you, Franz. Thank you, Christine.